believe it or not, I got $6 million from DARPA based on a science fiction vision of using 3D printing to create tensegrity-based materials that would be strong, flexible, resilient, and porous, to both create almost artificial skins to protect against biothreat agents. And then, so you're doing materials, medical, computer stuff. He said, nobody can compete with you if you can figure out how to harness the Boston-Cambridge region. He literally said that. I think America still has an edge for a long time if the government supports it and doesn't get in its way.
What is it? Fleming of St Mary's Hospital, London, who discovered that it produces the drug known as penicillin.
The question was, which drug would go with which cell?
It's a collaboration that's unexpected because it's between humans and the tiniest organisms.
The howl of a dire wolf.
Dorset lamb may represent a major landmark.
I did not start off planning to design proteins.
Scientific breakthrough will probably be something that hard to imagine.
Test at birth could map out an individual's entire future risk of disease.
Journey into science, right? So you're obviously a big shot now, but
big shot. Yeah.
When you were younger, what were you like?
I was exactly the same. That's what when I see friends 45 years out of college, and they say you're exactly the same. I basically, I got to Yale at age 17. I stayed there for 11 years. Went MDP, bachelor's, master's, MDP, then went to Harvard as a postdoc, stayed there for the last 41. I've never had a real job, I've been in the same kind of environment, and I've been able to, and I was breaking boundaries between disciplines by starting as an undergrad, having an experience in an art class that affected my science, and so I've always been doing that, and because I stayed as an undergrad at Yale for so long, I knew people in every grad school, I knew every library, so I did interdisciplinary pursuit of ideas from the beginning, and I've just kind of continued to do that till now. So at a personal level, I think I was very similar. Obviously, I'm a lot more grounded and confident in knowing how to do things, but as a personality-wise, pretty much the same.
If you had to take us back to the beginning of your sort of scientific journey, how did you get into it in the first place and sort of decide what field you were interested in?
Well, I remember, if you want to go to the beginning, I remember all my friends' parents for my birthdays would buy me scientific presents, and all I wanted were like Tinker Toys and Legos, you know what I mean? So I love to build, but I was actually got into Yale when I graduated high school. I was good in science, but I was really good in math. I was a mathlete, and I got the math award from a high school where there were almost 900 kids at the graduating class. And then I got to Yale, and I got advanced into the most sophisticated courses, but I looked at the kids around me, and they looked too nerdy for me at that point. And so I didn't really take science or math for the first year. I focused on humanities, which I loved. But when I really got into it, I think when I took developmental biology and watched how embryos form. I had a professor who was older and had some of the first movies of cells in a dish and things like that. I think that I grew up with my grandmother lived in the same house, and she had various health problems, and I was the one that I would take her blood pressure when I was a kid and things like that. So I think I always thought, I wanted to be an archaeologist when I was in high school, to be honest. It wasn't like I wanted to be a doctor, but I always was intrigued about that. I always wanted to learn anatomy. Like, I thought that was like a whole universe inside your body. So I was kind of pulled in that direction. But in college, I was able to major in molecular biophysics and biochemistry and do a combined bachelor's master's in a Bachelor of Arts and a Master of Arts, not a BS-MS, in molecular biophysics and biochemistry, because by doing the BA-MA, I could take courses in many other things. I already had a lot of advanced placement and so forth. So I was able to keep an interest in many different courses, and I just I love to learn. I love visual things, and somehow, my path that really triggered my career was taking molecular biophysics in molecular genetics in the 70s, before cloning had been developed, when molecular genetics was really molecular biology, molecular biochemistry, like how molecules work, the fact that DNA was a helix, and replication, the structure of viruses. And basically what we were learning was three-dimensional design, that basically it's the shape of molecules that dictates their function. Lock and key with enzyme substrate, the fact that it is a double helix that zips open and the 4 nucleotides. And I saw kids walking around Yale undergraduate campus with holding sculptures that looked like viruses that I was learning about in my class. Like, what is that? Where'd you learn to do this? They said, oh, it's an art class. I said, what class is that? It's called three-dimensional design. And so I actually have always loved art. I'm very visual, but I'd never done art, and I had tried to get into art classes at Yale, which was impossible if you were not an art major. You literally have to take interviews through the night. 3 a.m, I was interviewing for a drawing class, never got in. But I had a girlfriend who was a sculpture major, and by pure serendipity, her professor who taught the human body figure, taught three-dimensional design. His name was Erwin Hauer. So she brought me and introduced me to him and said, this is my friend Don, he's a scientist, he'd like to get in, take your course. And this is huge, big German, Austrian, imposing guy, like 6'4", like a football player. And he's like, why do you want to go to my class? And it was like, well, everything in biology is like, at the molecular level, is three-dimensional design. He said, what are you talking about? And I gave him the examples of, DNA is a helix, and it doesn't matter what it's made of, but it's the shape that's important. And he goes, and he says, tell me more. And so I would go, I was saying, viruses, I'm sorry, I went on and I basically said that, like, well, like your muscle is like actomyosin filaments, and they're sort of pulling by each other to create tension in your muscle. Because you're in. And then, like, I kept going on about viruses. He said, no, you're in, just come on Monday. And I took this course, and at the same time I was taking this course, I was just beginning to learn how to culture cells, doing cancer research across the campus in a medical school lab. And in the medical school lab, I was culturing cells for the first time. And if you've ever done cell culture, they grow on dishes, they're flattened. And then when they fill the dish and you want to move them to another dish, you add a enzyme, trypsin, and it clips their anchors, and they quickly ball up. And then you take them in solution and you put them on a new dish, and they spread out and flatten. Well, in this course, it was a very creative course, and the professor said, buy wood dowels and fishing line. So we all bought wood dowels and fishing line, and then he brought us to a wood shop, and we cut these wood dowels that were 3 feet long into 1 foot. We made little X's at the end so you could tie the fishing line to them. And then he brought us back to the room. There were probably 50 kids, and he said, all these sticks and strings on the table. He said, I'm going to leave you for the next few hours. I want you to build structures that are stable in 3D structures, but the sticks can't touch. And like for the first half hour, 45 minutes, we were just like looking at this stuff and trying to, tie it in different ways. And then some student who must have seen a sculpture by Kenneth Snelson built the first one of these tensegrities, tensional integrity, where the sticks don't string and it's pulled up by the tension or the forces within. Once one guy did it, like he saw every possible shape, like creative explosion across the room. And then he came back and he gave a slideshow about Buckminster Fuller, developed the geodesic dome, how this came out of his work. And he had a sculpture that was made out of these sticks and bungee cords, not fishing line, that were elastic. And it was round. And as he talked, he would flatten it and he let go and it would bounce off. And he would flatten it and bounce off. And I looked at that, and this was around 1975 or 6. The first papers had just come out on the cytoskeleton, actomyosin filaments in all cells. People thought they were in muscle, but not other cells, because they have the same density as EPON when you do electron microscopy, so they were invisible. But once they had fluorescent labels in about 75, every cell had these filaments that are sort of fibers and struts inside cells. So I went back to the cancer lab where we were working on a drug, and the drug caused the shape of this cell to change. And I said, oh, the tensegrity must have changed. And the postdoc said, what do you say? I said, the tensegrity must have changed. What's tensegrity? He said, well, I'm taking this art class and like Buckminster Fuller and sculptor. He said, never say that again. and basically I knew something was up, and that's when I started going to libraries. And I went to every library, physics, math, biology, art, because I knew them all at Yale. And I started to see that I had this, basically the idea that cells were built this way, that they had an internal tension and that stabilized their shape, and then got much bigger in the idea that physical forces are, could be as important as chemicals and genes. That was the dogma 100 years ago, 125, 50 years ago. But that was thrown out when chemistry and genetics came in. And this is another case of tipping point where it's either or, black or white. But the fact is there's always value in competing ideas. One might not be completely right, but there's probably something to it if a lot of people were interested. And so that led me to pursue the idea that, mechanical forces are important and that cells use tensegrity. And that was, by then I stayed at Yale as a MD PhD, and that became my path essentially.
I mean, that's absolutely fascinating. I think just one of the things that I've noticed is that art and sculpture and like an appreciation for an understanding of like the physical manifestation of a cell, like what it's like to be a cell, is lost, right? In
the
conventional
reductionist approach.
Exactly.
Yeah.
In this sort of modern
Same thing with
scientific
development.
teaching.
I remember when I learned developmental biology, we were watching all these cells pulling on each other and twisting and forming in 3D. And then in the 80s, it became the structure of the Hox gene promoter. That was developmental biology. So yeah, we went too far in the reduction, and basically when everybody was the power of genetics and everybody was moving in that direction, I was writing about collective behavior and looking at it as the system level, and so I was always kind of on the outside. People would curse at me at meetings about tensegrity. It was really kind of strange, but I was lucky to have mentors who, one of my mentors was George Pilati. He was the grandfather of cell biology, Nobel Prize winner. And, he would say, how is your tensegrity doing today? He was from Romania. And he gave me the book Synergetics by Buckminster Fuller, which he said, someone gave me this as a present for my Nobel Prize. I can't figure anything out of what he's saying, but maybe you can find it useful. And the first time my tensegrity models were ever shown in a scientific meeting, I found out later, were by Pilati, two other Nobel Prize winners. Jerry Edelman told me this later on at a meeting. And when I did my thesis defense, it was like, I have it written in his Cyrillic handwriting. Cells are tensegrity structures, of course, but what about this? What about that? And that was like an enormously... That gave me enormous confidence when I was then attacked, later on when I became a postdoc, and engineers would attack me saying, the cytoskeleton's under the membrane. There's no cytoskeleton inside the cell. And it's like, these guys work on red blood cells. They have no idea what a cell looks like. I'd done electron microscopy for years, And then later on, I worked with Judah Folkman as a postdoc. And Judah Folkman had been attacked about angiogenesis, the idea that cancer growth is dependent on capillary blood vessel growth. He'd been, attacked for years. And the fact that he stood up against these sorts of things gave me even more confidence. And I try to, give that kind of background of people I train now, because if you're not being criticized, you're not doing interesting work. You're not being provocative. You're not pushing the limits,
Mm-hmm. So obviously, you're most well known for the organ-on-the-chip technology.
Recently, yes.
More recently, the founding of the Wyss Institute. But sort of staying on the topic of tensegrity a little bit longer. Obviously, you were...
For a time, I was known by it. I mean, I was on television based on that. I was won, Esquire magazine something of the year because of that.
You're on Scientific American for a little bit.
And my Scientific American cover. So there are these waves in your career. And unless you are just channelized saying, I'm going to do one thing, but I had to keep growing. I'm not the type of person that can do one thing.
So to like a more layperson audience, what is this concept? Why is it relevant and important? And like, what applications does this have?
Well, first off, what really came out of the tensegrity model of the cell, and then showing that it actually is the way nature builds, so showing that molecules are built that way, and organelles are built that way, and cells and tissues in our whole body are built that way, where, if you see a skeleton in an anatomy lab, it's like, all these bones that are wired together hung from a stick. But it's not like us. We our muscles and tendons and fascia, connect those and pull them up against the force of gravity. And then it's the tension in my muscle, the tone, that determines the shape stability, whether this is rigid or flexible. That's tensegrity. Okay, that is like a fundamental property of these systems. So that led me to see that mechanical forces can affect biochemistry, because the biochemistry in our cells are immobilized on these support scaffolds, the cytoskeleton. And because of my early biophysics background, I knew that you change the shape of a molecule, you change its chemical activity. If you could do an ion channel, you can change, you could electrically stimulate it and change the opening or closing, but you could also mechanically do that. And so at one level, it's this idea that mechanical forces are important. That is now, being found to be true. With our organ chip work, we find this is true for, breathing motions affect your response to viral infection. Peristalsis affects inflammatory bowel disease progression. But it's also, tensegrity is being used by robotics people to build robots that are lightweight, flexible. People are building artificial materials that are more lifelike, that have tensegrity pre-stressed features. We've developed, and others develop, therapeutics that target this sort of mechanical signaling as an alternative way to treat disease. So it provides a context to understand how the body works that, for example, is relevant from physical therapists and surgeons to people who think they're working purely on chemistry, like someone working on inflammatory diseases, where it turns out mechanics is important as well. A simple example: first person who published papers on how acupuncture works came to me, quoted all my work, but she found that it's the twisting of the needles that pulls on the collagen in your connective tissue, that pulls on the integrin receptors, which we showed mediate mechanical force transmission to the cytoskeleton and changes in biochemistry, that results in changes in inflammatory response and changes in remodeling and things like that. So it's not obvious, but it's there everywhere.
Yeah, it seems to be very integral.
Yeah.
Let's talk a bit about the companies that have sort of spun out of this. Obviously, you've had Neomorphics, Tensegra. Talk to us about applications of this and sort of how they were commercialized.
I mean, the companies were sort of interesting stories. I mean, the first company I did was with Bob Langer, the great founder of companies of our generation, and Jay Vicanti. We all happened to be working in the Folkman department at that time, and this was in the 80s, when all you need is a minimal data to get funding. And the idea, that was the beginning of tissue engineering, basically. Jay Vicanti, who was a liver surgeon, and Bob had the idea of using biodegradable polymers that are, think of like felt type materials, and putting cells in them and essentially using those to make artificial tissues. They needed someone who knew biology, and so I came in as sort of the biological person. Dave Mooney, who's now a professor at Harvard, and Linda Griffith, Dave was the first grad student that worked with me during that time, and Bob, and then Linda was the first postdoc. So this helped sort of birth and train Tony Mikos, who's in Texas. Leaders in the tissue engineering field came out of that. Tensegra was originally molecular geodesics. That was in the around late 90s. Believe it or not, I got $6 million from DARPA based on a science fiction vision of using 3D printing to create tensegrity-based materials that would be strong, flexible, resilient, and porous, to both create almost artificial skins to protect against biothreat agents, and then medical devices. And we were using computer-assisted design, manufacturing through 3D printers, and engineering analysis. So we were developing really cool ways to design your optimal material for any application, surface area, anisotropy, meaning, directionality, mechanical load-bearing strength. For example, we worked on a lumbar disc for the back that was porous but flexible and would integrate, and then make it, and then even optimize it on the computer and then make it. And we actually had a device went into cows for the lumbar disc that looked really good. And then September 11th happened and everything fell apart. So that one died. It had become tensegrity by that point because it moved more to the orthopedics area.
Out of curiosity, what does this September 11th tragedy have to do with the...
Financial market. We were running low on money like every startup. There was a company that was talking to us about acquiring us or a partnership, and our money ran out just at that time, and when we went to the markets, everything was frozen. And then that company bought the IP at a yard sale price and just sat on it, basically.
As it happens.
So I've done a number. I mean, the more recently, Emulate is the big one. I'll say also I have. I'll talk about Emulate, but a company now called Cerulean. It was originally Free Flow, but that's a coating for medical devices that prevents blood clotting, working really incredibly well, and they're trying to move that towards the clinic. We had another company that was started out as sort of an artificial spleen, the idea of capturing pathogens from blood from patient who have sepsis. This is another tipping point story. Okay, this is sort of interesting. So sepsis is when you have, a microbial infection that leads to systemic inflammation. And almost all the therapeutics have failed, and they all target inflammation, part of this. But the fact is that there's a tipping point. In medical school, we learned that if you have a patient who you suspect has urinary tract infection, you take a urine sample, you spin it down, and you count the bugs, the microbes. If you have 100,000 and above, you treat them with antibiotics. If you have 99,000 and below, you just watch. There is truly a tipping point in nature at all levels, and it's just empirically, it's about that where it'll get out of control or your body will get it under control. So the idea was patients who have a systemic infection, if we could take the pathogens out, we may flip them that tipping point so now they can get it under control. And so we engineered at the Wyss Institute a capture molecule that binds over 100, if not more, way over 100 different pathogens of every type. Gram-negative positive bacteria, fungi, viruses, toxins. And we eventually immobilized it on a dialysis-like cartridge and basically take patient blood, pass it through. That actually went to first clinical study at 15 patients. It's safe, but that also died more recently in the last couple of years when the funding market went down. We're about to try to hopefully rejuvenate it. Another company is called StataDX. That's developing a handheld electrochemical sensor that my group developed that has a nano coating that prevents the background signal that you normally get that interferes with electrochemical sensors. That's really going well for multiple sclerosis and other neurological diseases where there's actually a biomarker now and the drugs are incredibly expensive. And to know whether the drug's working or not, to switch drugs, that's being advanced. There are other companies, I can't even remember what's going on now, but Emulate is the one on organ chips, and that one is one that is now, they're sold internationally. Almost every major, biotech pharma is exploring them. FDA has them on site. And that's the one that, I think it is beginning. It's one of the most impactful things I've done and most interesting to the public, because the whole idea of replacing animal testing, everybody loves.
So let's take a step back here and talk about sort of organ-on-a-chip technology. There seems to be a natural progression from sort of this tissue engineering work, this tensegrity approach to organ-on-a-chip technology. But sort of before we...
It's not obvious, though. It's a very interesting story. I've told this recently to various people interviewing me about the current, attack on science and let me describe this. I mean, economists interviewed.
Describe the technology itself first. Sort of like, what is organ-on-a-chip technology?
Organs on chips are small devices the size of a thumb drive that are optically clear, made out of a flexible rubber-like polymer that have engineered hollow channels, microfluidic channels that you can put fluids through that are less than a millimeter. We have two of them in parallel. You could think of like, the Holland Tunnel or something like that that are next to each other. But we have a porous membrane. And so we take living human cells from the tissues of patients or cadavers, and we can basically take different tissue type cells and put them adjacent and let them interface. So for example, the first chip was a lung chip where we did the air sac of the lung. So we had the lining cells of the lung, we had the capillary blood vessel cells on the opposite side, and we could flow nutrient medium through the capillary channel, like mimicking vascular perfusion, and we could put air in the other channel, mimicking air in your air sac. And then we have one trick we did, because of my belief that mechanical forces are important, is we had hollow channels on either side where we apply cyclic suction, and it's all flexible. So when you apply suction, the tissues stretch, like when you breathe in, and when you let go, it comes back. So we're able to mimic the degree of stretch and the frequency with breathing, which turned out to be all important. And to make an intestine chip, we take intestinal cells and we put them on one side and either capillary cells or connective tissue cells, and we mimic peristaltic-like motions. And we have trickling flow on the luminal side, and we have capillary flow on the other. And so that is an organ on chip. And basically what's amazing is that with high fidelity, it really recapitulates organ-level functionality. And I want to emphasize that it's an organ chip, not a tissue chip, because it has an organ or two or more different tissues that interface new functions emerge. Almost always a blood vessel is part of it to deliver oxygen, remove waste products, and so forth. And we have consistently found that having the physical forces of flow and matching breathing motions, peristalsis, stretching of skin, you need all that together to really recreate the functions that you see in vivo. And this is being used for drug development, for both drug safety, drug efficacy evaluation, but all drug discovery and personalized medicine.
There's something that I think a lot of people don't appreciate, which is that there's something about the mechanical force on the cytoskeleton that actually induces biomolecular changes.
It changes genetics, it changes biochemistry, absolutely.
So you need this to be able to recapitulate organ function.
And
it
impacts the development, so the differentiation, the specialization of the tissue depends on that environment as well. And that's how embryological development works. There's no artificial materials. The cells put out materials, they grow, they pull on each other, they define the growth patterns, and they continuously remodel. I mean, we had a paper years ago with how the tooth forms, and people had worked for years on all the genes involved and all the growth factors involved. And we basically found that, like, by squishing the mesenchyme, the connective tissue, that triggered the genes that everybody was looking at. And it actually had to be a physical event to click on the formation of tooth. And we could make artificial materials that would cause a similar compaction, and we could kind of click on a similar pathway.
Fascinating, yeah. And I guess like one of the interesting things is that even though there's been this general decrease in funding for science in the US, this particular area of science around replacing animal testing is getting a lot of attention. Why is it getting attention, and sort of what's the application?
Well, first off, it's interesting that, and I was involved talking to Congress, among others, about the FDA Modernization Act, which was, I think, 2022. In the last 5 years, I think this is one of the few things that was like entirely supported by both sides of the aisle. It was like Ron Paul on the Republican and I'm blanking out who it was on the Democratic side, Jeffries, I guess, I think it was, and there was unanimous support for the idea that the FDA needs to start thinking about human relevant models instead of animal models, because if FDA has published this, NIH, pharma, everyone knows that on average, 70% of the time, the results from animal studies are wrong when they get to clinical trials. So it's more often than not. As a grad student, would any grad student design their experiment based on failure? But this is the way a multi-billion-dollar industry has to do it, because they have to make the FDA happy and give them what they want. And in some areas, it's like 95% of the time it's wrong. And like in neurology, neurodegenerative disease, some cancers. So people have recognized there's a need to replace animal tests, but there were no alternatives. I think the work from organ chips and organoids, which are basically balls of cells grown in a gel that have stem cells that come from primary tissues or induced pluripotent stem cells, both of those have made it obvious that human models are actually now beginning to be available and in many ways better. The organ chips are really unique in that we can mimic, because we have flow, we could mimic the way drug levels change over time, what's called pharmacokinetics, why a doctor prescribes a drug once a day versus 3 times a day. Totally different efficacy and safety profiles with how the drugs change in your body over time. We could manipulate that in our chips because of flow. You can't do that in organoids or any conventional culture. When we do that, we mimic human responses that are known to be sensitive to drug regimens. We also can grow microbiome, which is like one of the big paradigm shifts in medicine in my career. When I was in med school, nobody ever talked about the idea that, there's microbes in your gut are important for health and disease, but now it's pretty well accepted. And if you put a complex mixture of microbes on your culture dish, static or organoid, in 16 hours, it'll die. It's contamination. Mm-hmm. With our chips, because we have flow and we have the physical motions, and the cells specialize so that the intestine cells or cervical cells put out mucus that's normally protective, and there's flow through the lumen so that some of the cells that are proliferating or microbes are washing out, so you get a dynamic equilibrium. We can culture them for days and look at like direct microbiome human interactions, and we're able to actually study the mechanisms of how this works rather than just look at correlations, metagenomics, or how the levels of bugs correlate with disease or health. And so that's been really interesting also.
The microbiome angle I didn't know about, which is absolutely fascinating. Wow. Okay, so clearly there is a need to correlate the assay better with sort of the clinical endpoint. And
let me add that there's one additional side, which is personalized medicine. All right. So we just published a paper I did with a surgeon in McGill, where he took patients who have esophageal cancer. He's a cancer surgeon. They almost always fail the first trial treatment, and they have to then go to another cancer chemotherapy, but they don't know which one to try. And so the question was, could you predict who would be resistant or sensitive to the next drugs? So he got organoids from their cancer. So that means like collecting the cells that are stem cell-like. He grows them in a gel, and then we break that up and we put them on one side of our chip, and we also got the connective tissue cells which interface with the tumor in the esophagus, and we put those on our chip. And we compare the responses to drugs in the organoid static dish versus where we flow the drugs like the patient would see them over a similar time frame and dose. And the organ chips were extremely good at predicting the patient responses, and he's now using that to select drugs for those patients. And so, whereas the organoids were not affected because they died when you give them direct exposure of the same dose of drug that a patient would see. So that's another use. It's not just to replace animal testing. And then finally, I think the game changer in drug development is right now, big pharma will spend hundreds of millions to develop a drug, tens of millions on a big clinical trial, and they almost always fail. And what they do is they have statisticians number crunch the results and say, was there a genetic subpopulation or a group with similar phenotype that responded better than the others? And if they find it, they'll do a narrow trial, and if it works, they get approved for a narrow application. With patient-derived cells and organ chips, you can imagine from the beginning, start out with a low number. Let's start out with 100 people, men a certain age who have a certain type of IBD, inflammatory bowel disease, and you have drugs that you're developing tested on them. Well, maybe 50 respond really well, the other 50 don't. And then take their liver chips and kidney chips to look for toxicities and find 35 who have no toxicity and respond well, and use them for a clinical trial. And then you sort of flip it on its head, faster, cheaper, more likelihood to succeed, quick. So it's more than just replacing animal testing.
Yeah, absolutely incredible. Yeah, so obviously, A, personalized medicine, B, improving the correlation between the assay and the clinical endpoints, and sort of C, removing animals from this chain.
D or E, you get insight into molecular mechanisms of disease and normal physiology. So get back to mechanical. We found that when I went to med school, I learned that if a patient has surgery with anesthesia, you want to get them on liquids very quickly to get their intestinal peristalsis moving again. Because if you don't, you can get ileus, which is overgrowth of bacteria in your gut. Now, all the textbooks say that's because flow stops. We used our chips. We found that it's the peristaltic mechanical deformations that control the bacterial overgrowth, because we could keep the flow and stop the mechanics. It was the mechanics. We also use artificial intelligence now in combination with these chips, in combination with data, genetic data available on the web from patients, to quickly repurpose drugs, meaning take an existing FDA-approved drug and find a new use for it. We did that during COVID. We've done that for Rett syndrome. And so there's, I like to say that the chips literally provide a window where you can look at molecular scale activities inside living cells, inside a human relevant tissue and organ context. We can visualize what's going on. We can measure the outflows and identify biomarkers of disease that would be blood biomarkers if we collect from the blood vessel channel, or stool biomarkers if we collected from the lumen of an intestine chip. So it's more than, there's a lot there that's not obvious.
Welcome to the Frameshifts podcast, where we spotlight the world's most ambitious biotech founders, investors, researchers, and changemakers. This episode is brought to you from the offices of Polsinelli, a national law firm that helps biotech companies do everything from incorporation to patents, trademarks, and venture financings. Thanks to Polsinelli for being part of the growing ecosystem of scientists, investors, and builders shaping the century of biology. Yeah, absolutely. So we've talked about tensegrity, we talked about organ-on-chip technology. The other big thing you're known for is the Wyss Institute.
Yes.
And obviously this is a paragon of sort of modern entrepreneurship-focused biomedical institutes. It's one of the best in the world. But I think a lot of people don't know about its origin story. Like you finally built this institute that's sort of more than the sum of its parts, and also more than what you could have ever achieved by yourself.
Absolutely.
Which is something to be proud of. But
I'm very proud of it. It's one of the most gratifying things I've ever done, actually. I mean, it started about almost 20 years ago. I literally got a letter from the provost asking me and Dave Mooney, who had been my grad student years before, to co-chair a committee to envision the future of bioengineering across Harvard and the hospitals, which, by the way, is an easy thing to say because the hospitals are separate competing institutions that compete with each other and with Harvard, but we all are Harvard faculty. But I learned soon after we started that there was a donor who was interested in maybe doing something big, and he had come to Harvard, and was introduced to all these famous scientists, but none of them knew what anybody else was doing. They were all in their own separate silos, and he was looking for really something disruptive. And I had, by chance, in years before that, written up 2 visions for institutes. One was in biomimicry, when I was doing molecular geodesics and tensegrity, which was bio-inspired, 3D printing of materials. And then later I was doing one, I did one on biocomplexity, because my group had gotten into sort of a physics-based approach to looking at gene regulatory networks and the idea of the way you switch cells between states, healthy, disease, growth, and differentiation, is by hitting multiple nodes in the network and switching attractor states, which comes from physics, rather than linear pathways. And that's actually where I first met George Church, because we were both on a committee where the Harvard MIT HST program was trying to think about garnering interest enough to have a research group in Harvard Medical School when they were building the new research building. And they were looking for a vision, and that's where I developed this Biocomplexity Institute. So I had these 2 things I had written, and we had this opportunity. I kind of merged those 2 ideas, and then Dave and I, we talked to someone who said the key to having a successful, enterprise that involves lots of people across multiple institutions is shoe leather, which meant like you got to go to people's offices and like just meet people. So Dave and I went in every school, other institutions in Boston, Cambridge, in the hospitals. We went to other institutions in Boston, Cambridge, because when I had my startup around 3D printing, it was funded by DARPA. I will never forget, my program manager said, you're working on computer-assisted design, manufacturing, you're trying to do biomedical, we work on, non-medical for pathogen protection. So you're doing materials, medical, computer stuff. He said, nobody can compete with you if you can figure out how to harness the Boston-Cambridge region. And he literally said that. So when we did this institute, I really argued that this can't just be Harvard, and really has to figure out how to leverage this whole region. The other thing that happened was the donor, when we met with the donor, I remember him saying that, he said, I know big companies. I'm the CEO of a major orthopedic implant company. I know big companies, like, they can't innovate. Like, I can't make my own people take risks or chances. They graded product development and marketing, but they can't innovate. It's very hard for them to innovate. He said, I see you guys in academia, you do innovative stuff, but you publish papers, nothing ever happens. And he wanted to see sort of a startup in the midst of the world's greatest academic environment that have near-term impact. And that was, putting all that together became the kernel of the Wyss Institute. Maybe 75% of it was sort of biocomplexity, biomimicry, materials. And then we started to, I brought in George Church and Jim Collins, all the people, George Whiteside, people I had collaborated with. I developed, wrote this vision concept with Dave that everybody loved. I mean, it was amazing. The more we presented, more people loved it, and the donor loved it. And so that was the beginning. How it actually got started is a long story, in the sense that the donor did due diligence on Harvard, and everybody told him it's all siloed, it's the wrong place to do this sort of thing. And so he kind of walked away for a couple of years. Harvard kind of gave up on him. I would just keep writing him emails that he never responded to for a year and a half, that basically said, we're still working together, we're still excited. And then I happen to have a house in Martha's Vineyard, and he does too. And I wrote him in August, and out of the blue, he wrote back saying, "Oh, I'm so sorry. It's been, I've been doing other things. Why don't you come by?" And we talked. He got, I showed him some things are going on. He was very excited. He said, let's talk when we get back, when you get back to Harvard in the fall, and he gave me his cell phone. He never contacted me in September, so I called him, and he was about to get on his plane in Switzerland, and he gave me the name of a local person who he said he trusts, and that guy came to meet with Dave and myself, and we said that we'd been talking to Wyss about, $125 million gift, and he laughed. He said, "Yeah, right." He said, "Well, let's start from scratch. I want to see a risk assessment." And so we said, "Great." And he left, and I looked at Dave, and I said, what's a risk assessment? He said, I don't know. And we put together a risk assessment that was scientific, technological, but it was mostly geographic, political, historical. It was like, we're worried about at a university having haves and have-nots and people being jealous. We were worried about if this come under control of a dean or two deans, there's just going to be fights for control. We were worried about we're all on separate campuses. We're, all things you would never think of. And the guy looked at it, he said, this is the best risk assessment I've ever seen. And I think we could work together. And we basically developed a business plan like we were doing a startup. And with phase-in plan budgets and all this thing. Harvard was very concerned, got very upset at me. We're not going to have faculty deal with a big donor. And I said, I'll talk to the donor. But he said he would only talk to Dave and me. And they knew he was for real. And I literally got the agreement signed in the Black Dog restaurant, and he was wearing shorts and a polo shirt, as was I, in Martha's Vineyard. I mean, it was kind of an amazing thing for $125 million.
This is incredible. Just like the way that things actually get done.
The way things actually happen. I always say life is a nonlinear process, I gave a class day graduation lecture at Dartmouth Medical School, and I said, life, in nature, there's a thing called a random walk, right? Molecules moving in space. But in biology, there's a biased random walk, right? With the macrophages, it's a random walk, but if he sees an infectious bug, he'll go back and forth, but over time, he'll go right to his target. He'll end up getting to the right place. And I said, that's my life path. And I think the real life path, it's not linear. You don't determine what you're going to do. But if you follow your passions, you somehow end up where you want to be. It's an amazing thing.
It is, yeah. I haven't lived long enough to arrive there, but I hope I do.
Somehow I bet the idea that you're doing interviewing and making things for the web was something that you always kind of wanted to do, but that's not what you thought you'd do when you were applying to grad schools, so it happens.
Absolutely. Okay, so in this journey of sort of the establishment of the Wyss Institute, it seems like this guy, I'm not sure if I can pronounce his name, Hans-Jörg Wyss.
Yeah, perfect.
He seemed to be ready to give money. He seemed to be wanting to do something big, as you mentioned. But is this how it usually is? Is it usually this this big shot sort of wants to do something big? Or
I think
I
think every case is different. I think first of all, what I think most scientists don't realize when they look for donors is like they're pitching what they do to a donor and hoping they're going to just throw money at them. And really what you're trying to do with a donor is try to understand what's their passion, what's their, how do they want to change the world, and see if you could do something that fits with what you want to do that will advance their vision and something that has not been done before. They don't want to know what you've done. They want to know what could do that's be disruptive if they gave you a huge amount of money. And we spoke that speak, and he loves disruptive. He likes shaking things up and making things, like showing the establishment there are better ways to doing things. Like he's done that his whole career. And the fact that, we were trying to do something that was more translation focused and even commercial focused at Harvard, that is notoriously wary of that. And it was for the first 10 years, they gave me they were constantly thinking I was going rogue, that it was going to be out of control. Because we're a separate 501c3. We're not in any school. I don't report to a dean. I report to a board of directors. And clearly Alan Garber, who's the president, was the chairman of the board at one point, and there are deans on it, but there are also venture capitalists on it, presidents of other institutions, deans at other institutions. So it was kind of giving up a little bit of the control that they normally had. And that was very hard in the early years for Harvard, especially when we started 2009, the economy crashed and their budget just dropped, and I just got $125 million. And because of that risk assessment that I had, we structured the governance to minimize those risks, including having control over finances. So when the deans when Harvard lost $8 billion in the endowment, and the dean said, can I use some of that money for graduate students? It's like, I'm sorry, but it's very clear what I can use the money for and what I can't use the money for, and I can't do that, and that saved us, actually.
Yeah. Hey, one of the through lines of, I think, your success and the success of this area is this and just like this great flywheel of American innovation
Yeah.
is this interplay between academia, between the federal government, philanthropic donors, and also the venture capital ecosystem, right? Obviously, this is, I mean, I think throughout history, this seems to be a moment where it's changing the most. Like all of these things seem to be falling apart. The funding environment for venture capitals is quite terrible.
Yeah.
Academia is struggling because the federal government is reducing
Strangling
budgets.
it, yes.
What does the future of biotech and academic funding in America look like?
You were saying, you made the comment that, in the past there was this alliance between, a partnership between academia, the government, and philanthropy. And it's really true in the sense that, so Hans-Jörg Wyss gave a huge amount of money to the Wyss Institute, but we doubled his investment by getting government funding and foundation funding. Conversely, we've gotten government money, but we more than doubled it by getting foundation funding and donor funding. You know what I mean? And so, the Trump administration, talks about that we have, we pay lower indirect costs for these foundations like the Gates Foundation. But again, they're amplifying the investment that they put into it. And the same thing with donors, and it really was this positive feedback loop of the openness of the government since the 1940s to realize that they want to create the way to create an innovation engine that's going to drive technology, that's going to drive the economy, because technologies drive the economy, is to attract the best and brightest from wherever they are and provide fuel through funding of education and research. And yeah, we're at a real concerning time where the current government, they're great at destruction. They're not good at creating or thinking strategically about. They think strategically about destroying and tearing things apart. They don't think strategically about building for the future. It's almost, well, let's eat the seed corn, and not worry about what happens after the administration. And that is terrifying, because my big concerns for the future are who's going to go into science when you see this sort of uncertainty. Very few Americans were going into science because of the salary levels for postdocs and the years it takes of training were abysmal compared to other fields. And the international students were attracted because this is where their lives can change. And America was so innovative, and they could grow in ways they couldn't grow in their own countries because they don't have the flexibility and ability to cross barriers and work interdisciplinary and do startups. And one thing so before I go into what I was going to say, I do worry about the future because American students are not going to go in, and then they're creating an incredibly challenging environment about visas, and so we're going to lose the young people. The young people are the future of science. I mean, they do the work in the labs. They come up, they really come up with the great ideas. We guide them. We'll throw ideas. We'll push them in a direction with an idea, but then they make it happen. And so that's what terrifies me the most about the future, even more than the funding. Mm-hmm. Funding will go up and down. You can find funding in different ways, but if you don't have the human capital, that is concerning. The story I wanted to say goes back to, we didn't cover how I got to organs on chips from tensegrity and all that stuff, but it also is directly relevant to what we're talking about now. The magazine The Economist interviewed me a couple of times when we got attacked by the Trump administration, and I lost $20 million through stop work orders overnight, within 5 hours after Harvard turned down the offer. And I said, I'm worried about a brain drain that's going to start, and now it has started. And she said, well, wouldn't that be a good thing for the rest of the world? And I said, it would be a good thing for the rest of the world, but you're not going to create the innovation engine that we see in Boston and Cambridge. This is a singularity. It's a critical mass of brilliance, of juxtaposed right next to each other. I've collaborated with Boston University, Tufts, medical school, all the hospitals across Harvard, because of juxtaposition. And we have venture capitalists in the same building. Third Rock Ventures, rated one of the most successful, is upstairs from us, and all these startups. And then we have the hospitals next door, and we have all the major pharmaceutical companies and medical device companies and IT companies. So, when you take away a student, and I mean a good young faculty, there'll be one great person at each place. You will not recreate that. Mm-hmm. The other thing is that what's driven our innovation engine is getting the best and brightest from around the world, who then stay here and become entrepreneurs and create new industries and new jobs. So she said, well, can you give me an example? And I thought back, and the example was organs on chips, actually. So I'd worked on cell shape, cell adhesion, mediating mechanical forces, and so in about 1990 maybe, I got an email from a grad student at MIT named Rahul Singhvi, who said, I was given your name because I need advice. I work on cells grown on microcarrier beads in a bioreactor for biomanufacturing. This is at MIT, okay? So I said, sure, I'll talk to anybody. So he came over, and I basically came up with the idea to change the surface topography of the microcarrier beads, maybe by etching them to make greater strength. They were popping off. And he said, well, it'd be hard to do the beads. How do we explore this? So he had access to a laser, and so we took glass slides, and I had to make lines of different spacing on the micrometer scale, let's say 100, 50, 25, and so forth, and in perpendicular directions, so you get different sized squares and rectangles. And then we coated it with matrix, and we plated cells. And we controlled the shape of the cells, it turned out, because they grow on the top of the mesas. We controlled the growth. We controlled their nuclear spreading and the growth, and that worked. That basically fit with like my whole idea that cell stretching would control biology. But he was then, and this is a true story, he was in a bar in Cambridge with other grad students from Harvard, and everybody was talking about what they're doing, and they say, oh, we have a much better way to do that. And George Whiteside's group had just developed soft lithography and microcontact printing as an inexpensive way to make microchips. And so they started to get together, and it turned out to be a much more effective way. That led to our first paper in Science in 1994, I'm sorry. That was really the beginnings of nanobiotechnology, controlling things on the nanometer scale, but using that to control biological functions in cells. And long story short, that grad student graduated, went to Merck. I hadn't heard about him for years. It's funny, I just met him recently through a White House-based thing. But he then ended up being, CEOs of Novavax during COVID that made vaccines. He's now got a major company that's competing with China in biomanufacturing. He's become this, leader and created industries, and that is like the way it works here. Not only did it lead to breakthrough science, it led to, jobs and economic leadership. That is Boston, Cambridge, like to a T.
Mm-hmm. America has 3% of the world's population, I believe, and it attracts almost a majority of the world's intellectual capital because of this promise of.
And we're giving it away on a silver platter to China, and then it's just they're laughing all the way to the bank. It's unbelievable. And what Americans don't realize, I said this in an interview recently, which is that years ago I heard that someone did interviews with kids, inner city kids, and asked, where do you get vegetables? And they said the supermarket. And they, and you ask Americans, like, where do you get drugs? Where do you get iPhones? It's like these big companies. They all came out of academia, and then startups, and then they got bought up by these big companies. Mm-hmm. And so that economic engine dies if you don't support the universities. And this current administration is trying to kill it every way it can, including the IP ownership, which is going to kill investment, the visas. I don't understand it because they're so into American leadership and finance. But so I don't know where the future of science lies, but my concerns are great.
I was hoping you had more answers.
I think you will always see innovators innovate. And I think there will always be places. I think philanthropy is really made a major impact on science in that during COVID, when most universities shut down, including most of Harvard, we were up and running at full speed, busier than we ever were, and we led to diagnostics, nasal swabs when there were none being manufactured, drugs, and it was the Wyss, it was the Broad, it was the Channing Zuckerberg. It were these institutes that had philanthropic backing, that had some hard money and some, all of us have people that have been in industry before to some degree. The Wyss maybe has a higher percentage of people who know how to like make things happen and translate. My sense is that it's going to shift more to applied research. But even when you do applied research, and I learned this from DARPA, I say we don't do problem-focused research, we do solution-focused research. There's an impossible problem. We will bring people who've never worked on it before together and come up with like total out-of-the-box ideas and make them a reality. But in the process, you have to do new science. You have to uncover new paths and insights. And so it's a different way of doing breakthrough science.
Part of the sense of loss that I have around a lot of international students, like no longer considering America their place to become trained and
or
to
be
build
safe.
companies.
Yeah.
One of the reasons I'm sad about that is because often they'll return to their home countries, but they won't have quite the engine of sort of economic productivity that allows them to bring their truest selves and their most ambitious selves to reality. And that's something that we lose. But then the sort of more provocative question is, and this is more of a devil's advocate argument, is it so bad if the economic engine of the world moves to China, if that becomes the destination for the greatest advancements in biotechnology?
America has the ability to innovate, and I think a lot of it is due to how we educate. The fact that we don't decide what we're going to do when we're 15 and get channelized down a narrow path. Most of the world, in Europe and in other places, they take tests at 15 and it's like, you're going to be a doctor, you're going to be a lawyer. The fact that I, could take art classes the same as science classes, the same as history, the same as, anything, really allowed me to realize that there are no boundaries between disciplines, and that, nature doesn't have artificial boundaries between, as Buckminster Fuller said, between, art, chemistry, physics. It's all one thing. And the fact that we can take risks and fail, and basically, if you fail with a startup, then you're a repeat entrepreneur when you try again. And so that's very American. And the fact that people will invest in things like that is very American, the whole venture thing. Now, I think it'd be good for the world for it to propagate out, and it is propagating out. China is so top down that I think it's going to compete on whatever exists now in ways we can compete. Mm-hmm. But I don't know if it's going to generate the next wave of things that we can't even conceive of right now. Mm-hmm. So I think it's good that the whole world changes in ways that could advance innovation and translation, but I think America still has an edge for a long time if the government supports it and doesn't get in its way.
Mm-hmm. Yeah, I've actually heard this before, and I think I agree with the sentiment that China as a nation can allocate substantial amounts of capital to sort of strategically important areas of innovation in a way that
and bring full force to bear on it, and that's where we can't compete because we're so internally competitive and lack. We do have so many regulations. Every company's going to come up with its own VHS or Betamax and fight for years rather than just doing one thing and rolling with it and making it work. So we have problems to compete, there's no doubt. But to come up with the next thing beyond AI, I'm not sure that's going to happen in China. But, it's changing because a lot of those people have trained here and internationally and have gone back. But having an ideological constraint on creative thinking, whether it's China or now here, holds things back. And I think we can compete with China if we go back to this open-ended support for innovation in its most elemental self,
Well, one of the ways of potentially addressing this is to sort of start more institutes similar to the Wyss, backed by philanthropic donors. And I'm wondering if you have a playbook in mind for would-be institute founders such as yourself.
Well, I have all these people who get large amounts of money contacting me and asking me, what I've learned and all these sorts of things. And unfortunately, most of them end up doing the same sort of thing, which is, say like when we started, we had measures of success which were very specific, obviously great people, but, intellectual property portfolio, startups, licensing agreements, and products in the pipeline in 5 years. It was very, when I ask these other people, you have measures of success, of course, to do great cancer research. Like, well, what does that mean? Like, or they structure it in an academic department and so forth, and they don't have independent governance and so forth. So, I mean, I think you need people who, when we started the Wyss Institute, I remember Harvard was very wary if I would hire people who kind of had a chip on their shoulder, like something to prove. That's not the type of people they would hire. I was only looking for people who had something to prove. All of the faculty were successful in their own areas, but they were mavericks. They were quirky. I mean, George Church is not your average geneticist, and Whitesides is not your average chemist, and I wasn't your average. I was in surgery and pathology. I mean, and so we all knew that we saw things differently than everybody else. We could do what they do as well as they could, but we thought we could do better because of our novel perspective. And each one of us had a different perspective and different, there's many types of genius and styles. And so that was another challenge in building the Institute, was not coming up with one culture, but actually the culture was to let different groups have creative freedom to work the way they work, which took me a while to fight my the board, actually, until they saw that was right. But I think I'm sure there are people out there that could be great leaders of institutes, but they have to have a vision and a passion and be hungry to change, and also really develop, like you do a startup, a business plan that actually has some coherence. So we didn't know exactly what we're going to do at all, but we had a vision, and we knew the areas, and we had people to start with, and those people all, had a common vision that they bought into. I've been on. We started a second institute in Geneva, and there they started. It was an interesting story. So, when we started the Wyss Institute, and he was going to give this gift, Dave and I were acting co-directors, and Dave didn't want to take on this administrative stuff, and this is like my vision, so like I wanted to do this, and Harvard wanted to use it to recruit some famous person who was a famous engineer to do this, and they told Wyss that, and he said, look, this is like investing in a startup. I found a great CEO. I love his vision. He pulled all these people together who are behind him. They're all behind him. I'm not going to give $125 million and tell him that his first job is to find his replacement. It's not going to happen. Mm-hmm. So he actually put into the founding documents that I had to be the founding director, and that was then in later ones. But when we did the Wyss Geneva, they didn't have a director, and it took 1.5 years to find somebody, and then he turned out to be somebody who didn't know the people, didn't understand the culture. So they're much slower in trying to get up and running, and it's years, and they're still trying to get up and running in a functional way. So it's not only the person who could be a scientist who could do something like this, it's also structuring governance and knowing your environment. If I were in a different place than Harvard, Boston, Cambridge, I'd probably be a different place, mm-hmm. different structure, mm-hmm. done differently. It's all about people. Mm. That's really the answer. It's all about the people, and you have to structure things around the people.
It's more than just throwing money and building a building and
there are a lot of places that got a lot of money and nothing came out of it. Mm-hmm. I mean, I'm very proud that we have 12 core faculty, all of whom have their home lab still. We have maybe 12, 14 other associates or even less involved. And we were responsible last year for, I think, over 30%, 28 to 35% of the startups, licensing agreements, IP submitted last year, all of Harvard University. That's FAS, HMS, engineering, public health, everything. It's amazing. And we've done that almost every year, for the last 10 years.
One of the things that drew me to the Wyss as a former startup founder was this motto that, like, if I forget the exact wording, but sort of if the work in your lab sort of never leaves the lab, it can never change the world,
it's
right?
not going to change the world until it gets out of the lab. Absolutely. Breakthrough discoveries,
Let's talk a bit about the faculty as well, and sort of, obviously an illustrious faculty.
Amazing.
Can you highlight some of the work within the Wyss?
George Church, one of the discoverers of CRISPR, founded so many companies, really DNA sequencing, DNA synthesis, innovations, genome engineering across the board, synthetic biology. Jim Collins was the founder of synthetic biology, and Jim is actually at MIT, but at the Wyss. And Dave Mooney in tissue engineering, and now, engineering the immune system, cancer vaccines. David Walt, founder of Illumina, which changed the world in gene sequencing, as well as Quanterix, single molecule diagnostics. Natalie Artsy is one of our young, one of our newer people, but now actually acting assistant director with me, associate director of the Wyss. She's developing all kinds of drug delivery systems that are amazing, from lipid nanoparticles to polymeric. We don't only do medical, we do sustainability as well. Pam Silver, synthetic biology, but also for environmental as well as medical. Joanna Eisenberg has done, many different things from, bio-inspired, biomimetic materials for preventing, coagulation, for purifying air, all just numerous different things. In the early days, we had bio-inspired robotics people like Rob Wood and Connor Walsh. Connor Walsh developed wearable exosuits that are not rigid like Terminator 4, but are Velcro and artificial muscles that are now commercialized and so FDA approved for people that have, post-stroke to help them walk normally. It's really, sort of the most brilliant and quirkiest across Harvard, Cambridge, Sangeeta Bhatia at MIT, Chris Chen at BU. Really amazing.
It is incredible, and it's I think a lot of it comes down. A very small part comes down to the design of the entrance, just like seeing all the companies on this big sort of big
screen.
wall. It's an inspiring place to be and to walk into.
Thank you.
Right?
It is. When we designed our new site, the architects wanted us to have our entrance on the 5th floor, and it would look like this law office, literally. I said, and they were going to have they said there's a big support beam that's diagonal that basically they said behind it they would put like a conference room or something because it was like a dead space. But that was the floor that opens up to a huge exterior deck that we had access to, and the whole building has access to. So I put our entrance there with windows with that plasma screen so that everyone could see what's going on, and visitors to those people can see. And then we exposed that big diagonal support beam, which is huge, because everybody notices it, and it's like, oh, that's cool. It's funny. You have to flip on the head oftentimes the way people view things.
Especially if
because
you're trying to
they do it
different.
Conventionally. They do it the way they've done it before.
Don, obviously, it's been a pleasure having you on. I'm going to switch to a quick fire round before we wrap up. So just kind of come up with the first thing that comes to your mind in response to the questions.
Fire round, okay.
Favorite place to visit in Boston?
Favorite place to visit in Boston? That's a good one. I guess the Common, because I live downtown. We walk to the Common. Yeah, and it's beautiful this time of year.
What's your favorite method for clearing your head?
Gardening.
Really?
Yeah, it's like either gardening or working with my hands. I'm helping my son renovate an old house, and like when you're planting, gardening, that's all you're doing. It's very mindful.
What is an experiment that went terribly wrong that you look back on comedically?
When I was an undergrad, I had an idea for basically 2D separation of molecules using sort of gel electrophoresis in one direction and electrophoresis in the other. And I convinced my faculty advisor to let me build this. But it was this huge thing made out of lucite. It was like an inch thick, filled with Cephedex beads, and it was just totally crazy. But the idea was right. But it was goofy, and I'd be in the cold room for hours trying to put colored dyes and show that it worked. Yeah, so that never really worked.
Got it. And last question, what's a book that you would recommend to our audience? Something that might inspire them?
There's for me, I mean, there are many, but the book On Growth and Form by Darcy Thompson from early 1900s, describing, before we had chemistry and genetics, how natural forms are created and developed, really inspired me. He really had insights, and he was the one who said that patterns in nature are diagrams of underlying forces, which was obviously inspirational for me, and I think is absolutely true.
Okay, we'll make a note of it. Well done. Obviously, it's been an honor to interview you, and thank you for coming on.
Thank you for